Okay, very good morning, my dear friends. Am I audible?
Yes, sir.
Yeah. So, welcome today's session. Today, most important topic that is meiotic cell division. It is most important topic in cell division, especially cell cycle. You may get 2 to 3 questions in cytology or cell biology. So that's why, my dear friends, listen carefully. Madam is going to explain in a wonderful way with beautiful slides. And if you have any doubt during the class, please clarify your doubts. So, in this regard, we would like to invite today's resource person, eminent personality, Dr. Pamuraju Manjula Madam Garu, Assistant Professor, becoming Associate Professor. Hearty welcome, Madam. She is working as Assistant Professor, RBVRR Women's College, Autonomous, Hyderabad. Madam, please welcome. Share your screen. Start your session, Madam. Please welcome.
Okay, thank you, sir. Sir, is it visible screen?
Yes, Madam.
Okay, sir. So today's good evening, one and all. So today's topic is 1 minute. Today's topic is meiosis. Okay, so meiosis generally cell division, two types of cell division, that is the mitosis and meiosis. So generally mitosis which takes place in the vegetative cells, whereas the meiosis which takes place in the reproductive cells. So meiosis, it is a special type of cell division which is necessary for the sexual reproduction. So meiosis begins with one diploid cell and containing 2 copies of each chromosome. Okay, so one from the organism, that is mother, and one from its father. Produces for haploid cells which containing 1 copy of each chromosome. So generally meiosis which takes place in the reproductive cells. So during the meiosis, during this process, so here it contains 2 copies of the chromosome. It is called as a diploid, and one from the mother and one from its father. So it produces the haploid cells containing the 1 copy of each chromosome. So in the meiosis, here 4 haploid cells will be produced. So history about this meiosis. So meiosis was first discovered and described for the first time in sea urchin eggs in 1876 by the German biologist, that is Oscar Hedwig, and it was described again in 1883 at the level of chromosomes by the Belgian zoologist Benedin in Oscaris worms egg. So next, the term meiosis was coined by Farmer and Moore in 1905. So first discovered by Hedwig, and later it was again rediscovered by Benedin in Oscaris worms egg, and the term meiosis was coined by Farmer and Moore in 1905. So generally where it occurs means, so generally meiosis which occurs in the eukaryotic life cycle which involving the sexual reproduction. So it always occurs in the reproductive cells only. Meiosis occurs in reproductive cells, whereas mitosis which occurs in the vegetative cells. So in lower plants, meiosis occurs after fertilization in zygote, and in higher plants it occurs before fertilization in the time of gamete formation. So what are the main phases of meiosis? So here meiosis again mainly comprises of 2 stages, that is meiosis 1 and meiosis 2. So again in the meiosis 1, prophase 1, metaphase 1, anaphase 1 and telophase 1. Again in the prophase 1, so again 5 stages are there, that is the leptotin stage, zygotin stage, pachytin, diplotin and diakinesis. Five stages are seen in the prophase 1. So it is the longest one. So next metaphase 1, anaphase 1 and telophase 1. Again it is followed by the prophase meiosis 2, that is prophase 2, metaphase 2, anaphase 2 and telophase 2. So meiosis 2, it is the similar to the mitotic division. Here it producing the 2 haploid cells to produce the 4 haploid daughter cells. So generally meiosis 2, it is similar to the mitotic division. So next one is, so what are the important stages in this prophase 1? So what are the what happening in this prophase? So during the prophase 1, the DNA is exchanged between the homologous chromosomes in a process called as the homologous recombination. So this often results in the chromosomal crossover. So in prophase 1, so crossing over and the paired and replicated chromosomes are called as bivalents or tetrads, and process of pairing the homologous chromosomes called synapsis. And at this stage, so non-sister chromatids may cross over it at a point called as a chiasmata. So these all are occurring in the prophase 1 of different stages of this prophase 1. Okay, so main one is exchange of the genetic material will occurs here. And that is called as a homologous recombination and form and replicate, that is the paired and replicated chromosomes called bivalents or tetrads, synapsis and chiasmata formation will occur in this prophase 1. So main events are occurring in this prophase 1. So first one is in prophase 1, the first one is the leptotin stage. So in leptotin stage, so the first stage of the prophase 1 is a leptotin stage, and in leptotin, that is Greek, leptonema means thin threads. So during this stage, the individual chromosomes begin to condense into long strands with the nucleus. So here they becomes, so thin thread-like chromosomes, they becomes condensed. So however, the two sister chromatids are still so tightly bound that they are indistinguishable from one another. So they are tightly bound here, two sister chromosomes. So next one is, so here thread-like, so leptonema means thin thread-like, and here the individual chromosomes begin to condense. They come closer and into the long strands within the nucleus. So like this. So these are thread-like chromosomes. So next one, in zygote. In zygote, zygote means paired threads. So the zygote occurs as the chromosomes approximately line up with each other into homologous chromosomes. And the combined homologous chromosomes are said to be the bivalent. So here bivalence formation will occur here. And in pachytene, so in pachytene, the homologous chromosomes become much more closely associated, and this process is called as the synapsis, that is pairing of homologous chromosomes. It is called as the synapsis. So this synapsis which occurs in the pachytene stage. So this is important point. And the synapsed homologous pair of chromosomes, it is called as the tetrad, because it consists of 4 chromatids. So here, when we observe this, so here these are the 4 tetrads. 4 chromatids are present here. So it cannot be observed until the next stage, but the synapsed chromosomes may undergo crossing over in the pachytene. So in pachytene stage generally, that is crossing over will occur. Synapsis, formation of synapsis and crossing over. So the chromosomes continue to condenses, they come closer, and in diplotin, that is 2 threads. So in this stage, the crossing over takes place and the homologous chromosomes separate from one another little. So nuclear membrane and nucleolus begin to disappear. So in diplotin stage, that is nuclear membrane and nucleolus begin to disappear, and here that is crossing over is taking place in this stage. Whereas in pachytene stage, that is the synapsis means pairing of homologous chromosomes will takes place in pachytene stage. And here the exchange of genetic material which takes place during the diplotin, that is crossing over, exchange of genetic material between homologous chromosomes. Next one is diakinesis. So chromosomes condenses further during the diakinesis stage, and in this stage the homologous chromosomes separate further and chiasma terminalize due to the contraction of the tetrad. So here the formation of chiasma, that is again they terminalize. So it is again they move towards their own side, and spindle fiber begins to form here. So here, when we observe this diakinesis, so here they will be separated. Okay, after the exchange of the genetic material, so it will be separated. So during this stage, the segment of one segment of the chromosome segment is attached to the another chromatid. So here in this way, here the exchange of the genetic material will be occurring here. So crossing over means, so here when we observe this crossing over, it is a process by which two chromosomes of the homologous pair exchange equal segment with each other. So here these are the two chromosomes. So here they are, here this one, this one it is called as a chiasmata. So in this place only exchange of the genetic material will taking place. So when we observe this, so here this material will be attached here, and here this material will be, same segment will be attached here. So it is called as the exchange of the genetic material, that is the crossing over. So importance of this crossing over is to produce the new combinations of the traits, and helps in the mapping of the chromosomes and selection of useful recombinants. So recombinants means formation of new, here new variant will be produced. So due to the exchange of this chromosome. So this is about the first stage, that is a prophase 1. So the main events are leptotin. First one is the leptotin. So here the leptotin, the main events which are occurring in the leptotin is the, so here the chromosomes becomes uncoiled and appear as a thread-like shape. So the centriole duplicates, and each daughter centriole migrates towards the opposite poles of the cell. So here it is called as a bouquet formation is observed during this stage. So here the thread-like, so chromosomes are thread-like and it appears like a bouquet. So bouquet formation which is seen in the leptotin stage. So next one is in zygote stage. The main events are, so the pairing of the homologous chromosomes will takes place in this stage, and the pairing of the homologous chromosomes, it is called as the synapsis. Okay, so the synapsis begins at one or more points along the length of the whole. So here this may be at one point, or it may occur at different points, more than 2 points also. So the synapses are pairing, which leads to the formation of bivalents. So the development of the synaptonemal complex. So in zygote stage, formation of synaptonemal complex will occur. So next in pachytene. So in pachytene, the pair of chromosomes become twisted and cannot be distinguished separately. So as a consequence of this synapsis, that is number of observable chromosomes is only half of the somatic number. So here it is called as a bivalent. So bivalent formation. Okay. So next one is so here only that is crossing over also takes place in pachytene stage, and it involves mutual exchange of the hereditary material. Exchange of the genetic material will takes place here. So in diplotin stage, that is homologous chromosome starts moving away from the each other. So during the separation of homologous chromosomes, so each bivalent chromosomes are attached with each other at one or more points. It is known as the chiasma. Chiasma formation will be observed here. So next one is in diakinesis. So the bivalent chromosomes become more condensed, and nucleolus separates from the nucleolar organizer and finally disappears. So chiasma move towards the ends of the chromosome. So it is called as the chiasma terminalization will occurs in this diakinesis. So these are the main events in prophase 1. So crossing over is important. So generally crossing over which occurs in pachytene stage. So next one is a metaphase. So metaphase 1, it is the second phase of the meiosis, and the spindle fibers organized between two poles and get attached to the centromere of the chromosome. So here the chromosomes moves to equator. So same like that. So here the spindle fibers are organized between the two poles. So this formation of spindle fibers, and they are organized between these two poles, and they get attached to the centromere of the chromosome here. Spindle fibers. Next one is the anaphase. So the anaphase 1 begins when the two chromosomes of each bivalent separate and starts moving towards the opposite poles. So in this stage, the sister chromatids remain attached at their centromere and move together towards the poles. So next one is the telophase. The homologous chromosome pairs reach the poles of the cell, and the homologous chromosome pairs complete their migration to the two poles. So a nuclear envelope forms around the each chromosome set, and the spindle disappears and cytokinesis follows. So during this phase, here two chromosomes. Okay. So centromere with centriole will end. Here the nucleolus reappears, and constriction of the cell membrane will occur here, and nuclear membrane reformed in telophase. So here, next one is the prophase. So meiosis begins without any further replication of the chromosomes. So the nuclear envelope breakdown and the spindle apparatus forms. So in the again, so it is in here in first one, that is in meiosis 1, that is here the formation of two bivalents will be taking place. Okay. So in prophase in meiosis 2, that is in prophase 2. So meiosis 2 also contains 4 stages, that is the prophase 2, metaphase 2, anaphase 2, and telophase 2. So in prophase 2, the meiosis 2 begins without any further replication of the chromosomes, and the nuclear envelopes break down, and the spindle apparatus forms, and the diodes chromosomes become thicker and shorter, and the nuclear membrane and nucleolus disappears, and spindle fiber starts to form. So it is similar to that of mitosis. That is meiosis 2, it is similar to the mitosis. So here also same nuclear membrane and nucleolus will be disappearing, and the diodes chromosomes becomes thicker and shorter, and spindle fiber starts to form here. So metaphase 2, second one. So centromeres are arranged in a line called as the equatorial plate of invisible spindle. So here in metaphase 2, that is the spindle operate, they are here, the metaphase plate will be formed here, and they will move towards the this equator. So anaphase 2. So the centromeres of each chromosomes divide, and sister chromatids separate to form the two daughter chromosomes. So spindle fiber contracts and pull the daughter chromosomes apart towards the opposite poles in anaphase 2. So in telophase 2, that is nuclear envelope forms around each set of chromosome, and nucleolus appears in each nucleus, and chromosomes, each cell, and chromosomes elongates to form the thin network of chromatin. So nuclear membrane and nucleolus reappear. So at last here the 4 haploid set of chromosomes will be produced. So here this meiosis 2, it is similar to the mitosis 1, whereas the some main events which are occurring in the prophase 1 only. So chiasma, crossing over, synapses. So these all are occurring in prophase 1. So this is the important stage, prophase 1. So what is the importance of this meiosis means? So produces haploid gametes so that the diploid number of the species remain constant generation after generation. So next one is, it is the source of genetic variation because crossing over brings together new gene combinations on the chromosome. So here mainly, here in meiosis, that is the production of haploid gametes will be produced, haploid cells only. Okay. So next one is the recombination, gene recombinations occurring in this meiosis. So conclusion, so meiosis, it is the reductional cell division which occurs in the germ cells and producing the 4 cells of half chromosome number from that of the parent cell. So nucleus divides twice in meiosis 1 and meiosis 2. And meiosis 1 involves the separation of the homologous, that is the reduction division, while in meiosis 2, separation of the chromatids occurs, that is equational division. So reduction division in meiosis 1 and equational division in meiosis 2. To maintain the chromosome number constant in a sexually reproducing species, meiosis is essential. So otherwise what happens? So the chromosome number will be increases. So this is about the meiosis. So the main events are, that is crossing over, chiasma formation, and synapses. So these are, so mainly in prophase 1 only, that is the exchange of the genetic material will be taking place. So this is about the meiosis, and next one is the differences between